Okay, Catabolite repression. I mentioned earlier in the previous video, the lac operon is switched on in the presence of lactose. But what if both sugars are available, glucose and lactose? You will find that in the presence of glucose, lactose do not get metabolized. That means even though in the presence of lactose, it does not get switched on. So what's going on? So here we look at a phenomenon called catabolite repression. It says that catabolite repression means as long as you have glucose, even if you have lactose, it will not be able to switch on like operon. I won't say that it contradicts this statement. This statement says only expressed independent of lactose, but you may add to this statement. It says lac genes are only expressed in the presence of lactose and absence of glucose. Right? If in the presence of lactose, if you have glucose present, it's still not going to switch on. How? Look at this. Okay. Actually, okay. It says. Let's read this. It says there is a molecule called the CAMP. CAMP stands for cyclic adenosine monophosphate. Right? Cyclic AMP. It's an effector molecule. How do you get this? Is because you convert your ATP. You've heard of ATP, correct? Adenosine triphosphate. Adenosine triphosphate, under the action of this enzyme called adenylate cyclase, is broken down. ATP is broken down into CAMP. Now, this CAMP binds to CAP. CAP is called Catabolite activated protein. Sometimes CAP in some books is also called the CRP, Catabolite response protein. Now, what happens here is this. If glucose is present, if you still have glucose, remember we are talking about a situation where you have both glucose and lactose. Now, if glucose is present, glucose also blocks the function of adenylate cyclase. That means to say, when glucose is present, you don't get CAMP. Why is CAMP important? CAMP is important because it needs to bind to CAP. When CAMP level is low, no CAP binding and no capsamin, therefore no activation. Now, okay, this is a better, clearer picture. Now, look at this. All right, these are four situations. Here, CAMP present, lactose present. All right. CAMP present, lactose absent, CAMP absent, lactose absent, and CAMP present, lactose present. Now, what's the difference here is that what happened in order for you to get transcription, you in the lac operon, not only it requires the presence of the inducer allolactose, you actually also need CAMP. If you get this CAMP binding to CAP and you have the repressor removed, then you get transcription. If you have this situation, how can you get this situation? You have CAMP, but you don't. The repressor is there. How do you get this situation? This situation, you will get this if you don't have glucose. How do I know that? Because I know if I have glucose, then you won't get CAMP. The fact that you have CAMP, that means glucose is not there. But at the same time, lactose is also not there. Because why? How do I know that? If lactose is there, then you get allolactose, right? So because you don't have both, so there's no transcription. What about this one? You have allolactose, that means you have lactose being broken down, but there's no CAMP. You don't have CAMP, so it cannot bind to CAP, it cannot bind to the cap site, so that means there's no transcription. Why does that happen? Because glucose is present, right? Glucose is there, so that means it blocks adenylate cyclase. ATP is not converted to CAMP. Because that you don't have CAMP. So although the repressor is removed, it still cannot be switched on because this cap protein that hasn't bind to that region. Of course, in this last example, both are not present. The allolactose is not present, the CMP, of course, is switched off. Okay, so this catabolite repression allows the system to choose glucose over lactose if both are present. Okay, so bottom line or take home message is that in the lactose operon, it is not only governed by the presence of lactose, which produces allolactose, which is an inducer to remove the repressor. It also requires the absence of glucose, because absence of glucose allows ATP to be converted to CAMP. Then only it can bind to the cap protein, which will bind to the cap site. And the presence of both and these two situations, again, presence of lactose, absence of glucose allows transcription. Okay, so that is catabolite repression. One question. This is like a chicken and egg situation. We learned that lactose operon is switched on, right? So that lactose can be broken down to allolactose. But in order for lactose operon to switch on, you need allolactose. So which one comes first? Let me ask you again. You need to switch on lactose to get beta-galactosidase to break down lactose to allolactose. But in order to switch on lactose, you need allolactose. It's a chicken and egg. So the answer is this. In gene expression terms, when you say the gene is switched off, it doesn't necessarily mean total switching off, that means zero product. When switch on, you have 100%. No, it doesn't mean like that. When you talk about gene expression, well, in microbes, in humans and so on, normally what you're saying is that you have low level of transcription against activated levels of transcription. So generally, most genes switching off or switched off means there are some very basal level of expression activity. You have very small amounts of beta-galactosidase being produced. But in the presence of lactose, then it starts to break down, and then you get allolactose, and then this allolactose will have a positive feedback, and then you break down further, and then so it gets with more lactose present, then it becomes higher and higher and higher and higher and higher. So that is what you mean by activation of genes. Switching off means like it just, from a lot of activity, it just slows it down to very, very minimal. Rarely genes are totally switched off, like you don't have any product, right? So I hope you can understand that point, and ask me questions if you find it difficult to understand. Okay, I'll see you in the next slide.